Hello, everybody who has just joined us. My name is Jennifer Summers, and I am the Program Development Specialist at the Wisconsin Center for Wildlife. And I would like to thank you for joining our seminar series today. This is the final seminar in our Daniel O. Trainer Wildlife Health Seminar Series. So thank you for everyone who has joined us for past seminars and for this seminar as well. First of all, I would like to say thank you to everyone who has helped organize this seminar series. Dr. Dubé, or Shelley Dubé, one of our wildlife faculty, has been critical in organizing this, as well as Marie Perkins, who's another one of our wild faculty. And also Ben Senninger helped us coordinate with a couple of speakers as well. And he's our waterfowl specialist on campus. And then this is hosted by the Wisconsin Center for Wildlife and through the College of Natural Resources at UW-Stevens Point. And also we are part of an UW extension at University of Wisconsin-Madison. So thank you to all of those people and all of those organizations for helping make this possible. And next, this is something in agreement we have with our local tribal communities, and something we like to do with every public gathering that we have, including virtual public gatherings. So we recognize the University of Wisconsin-Stevens Point occupies lands of the Ho-Chunk and Menominee people. Please take a moment to acknowledge and honor the ancestral Ho-Chunk and Menominee land and the sacred land of all Indigenous peoples. So as I said, this seminar presented today is the final seminar in our Daniel O. Trainer Wildlife Health Seminar Series. We had a total of 5 speakers, and all of those speakers will be available, or most of those presentations will be available on our website at uwsp.edu/WCW. There will be a link to the seminar series where you can watch the recordings of all the past seminars if you are interested. And then Shelley will, before I pass it along to Shelley, I just wanted to say to the participants, please, if you have any questions during the presentation, feel free to ask through the chat feature. So post your questions in the chat at any time. We'll read them off on the end. And then please, if you could, save on bandwidth and so forth, to keep your videos off. So thank you very much.
Great. Excuse me. Thank you for asking me to honor Dr. Trainer. It's really amazing to walk into a building every day that's named after one of my wildlife health heroes. I was in graduate school in the 90s, and a lot of the seminal work that I was reading and using in my research on deer were authored by Dan Trainer in the Midwest, and I'm a Midwesterner myself. But what we don't know, just from reading somebody's prepared work, is what kind of a human being, they really are. And Dr. Trainer was, first of all, an absolute card. He was a really, really funny, gregarious person, always had a smile on his face. He was also a decorated teacher, which makes him a great fit for UW-Stevens Point, since teaching is what we focus primarily on. And so the background that he had, which was a wealth of wildlife health knowledge and experience, and then a very unique set of soft skills or people skills that rendered him an extraordinarily effective first dean to our college. So with that, I will let Marie take over.
Thanks, Shelley. I'm really happy to be here today to introduce everyone to Dr. Jessica Head. I met Jessica back in 2012 when we shared a research laboratory space at the University of Michigan. And after that, we both transitioned over to McGill University, which is in Montreal, Quebec, which is where Jessica grew up. Jessica went on to get a professor position at McGill while I was doing my PhD work there. Jessica served as a committee member on my thesis research project, and I really found her to be such a wonderful mentor and role model, as well as colleague. We have a lot of similar research interests. Jessica's going to talk to you today about her research looking at contaminants on fish, but she also looks at the health effects of contaminants on birds as well. I'm really happy to hear Jessica's talk today and hear a bit more about her research on fish. So with that, I will let Jessica take it away.
Great, so I'll share my screen. How's that? Can everyone see the slide? Yep, good, I got a thumbs up, awesome. All right, well, thank you for the introduction, Marie. It was nice to hear a little bit about the namesake of the seminar series as well. And thank you so much for the invitation to present here. We were just discussing before the seminar started that it would be nice to be in person and actually get to meet people face to face, but it's certainly convenient to do it over Zoom. So there's always some positives. So today I'm going to tell you about an endangered species of fish called the Copper Redhorse that's endemic to Quebec, and our work to try to understand the role of agricultural pesticides in contributing to the endangered status of this fish. And on this title slide, I have Hugo Marchand listed first because this is really his PhD research, so he did all the work here. And in fact, other than the introduction, a lot of the slides are his, directly from his presentation, so I would be remiss to not give him credit. Sorry. There we go. Okay, so as I'm sure you all know, we're in the midst of a global extinction crisis. It's been estimated that 26% of mammals, 14% of birds, and 8% of bony fishes are threatened. And this is according to the IUCN Red List. What are the drivers? What are the factors that are causing this extinction crisis? Well, you know, they can be listed in different ways, but often you'll see habitat loss, invasive species, pollution, pathogens and disease, and overconsumption as factors that contribute to populations of wildlife being under threat. And the research that we do in our lab really focuses on trying to tease out pollution from the midst of all of these multiple factors. And that's what the talk is about today. Not on tigers, unfortunately, or maybe fortunately, but on a species of fish, as I mentioned. But before I get into that too much, I just kind of wanted to introduce myself and my lab, since, you know, especially since we won't be meeting face to face. So here's like the classic lab photo 2020 style, where we're all in our little separate offices or home offices. This here at top left is Hugo. So he's the one who is the PhD student who's really concentrated his thesis on this work. And Emily Boulanger also, who's was a master's student, is now a technician in my lab, contributed a lot to the design and the implementation of these studies as well. Our lab is, as Marie said, in McGill University in Montreal, Canada. I thought this was the perfect map because it shows you guys over here in Wisconsin and Michigan, where Marie and I met, but then also Montreal, which is an island here in the St. Lawrence, and also our study site, the Richelieu River here, which flows between Lake Champlain in the States and then empties into the St. Lawrence River and then out to the Atlantic. So I'll get back to that in a little bit, but for now, on the island of Montreal, McGill campus is located right downtown. That's this top photo here. Doesn't look like it's downtown because you're looking at the campus and then what we call it the mountain. It's more like a hill in the background, which is a large urban park, but behind you, you would have downtown in this photo. And then the lower photo is my campus, which is on the western tip of the island, the McDonald campus. And we're the Faculty of Agriculture and Environmental Science. There's a farm on campus, a beautiful arboretum, and we're surrounded by water, the Ottawa and the St. Lawrence rivers. So it's a really special place to be. And I live just off the edge of this photo on the bottom. So we're at McGill, the McDonald campus, and the research that we do in my lab is in the field of ecotoxicology. So studying the effects of environmental contaminants. And we work in my lab primarily on fish and birds, generally using early life stage models. So with birds, we inject chemicals into the eggs, or we study concentrations of chemicals naturally deposited into the eggs of wild birds, and look at the effects on the developing embryo. We also do multi-generational and transgenerational studies where we're very interested in epigenetic mechanisms and how the environment in the egg as the bird or the fish is developing, in the presence of contaminants and other stressors, how that can affect the individual not only during development, but also throughout their lives. And then even into future generations. And there'll be a little bit of that type of stuff at the very end of my talk today. We also work, in addition to lab models, so with fish, it's mainly zebrafish, and birds, we use chickens and Japanese quail. We work with wildlife in the field, and this is where we get into more realistic situations with multiple stressors, multiple contaminants. And then bringing it all together, we do a lot of molecular work. Lately, it's been a lot of transcriptomics. So using gene expression, looking at all of the expression of all of the genes of the genome, how many of them are turned on or off and at what levels, to try and inform our work on these early life stage models and the wildlife models. And in this project that I'm presenting to you, it really represents all three of these kind of different areas of research that we do in our lab. We do this work, as many ecotoxicologists do, under a molecules to ecosystems approach. So the idea here is that understanding the chemistry, so what contaminants are present in the biological tissues that we're studying, and the molecular biology, so those first level responses to the contaminants, the molecular, the cellular level, that information can really inform us about what's going on in organs, tissues, organs, individuals, and then eventually impacts that can affect populations and ecosystems. Ultimately, that's what we're interested in, what's happening at the level of population. But really, in order to make a connection between the presence of the contaminant and the adverse effect at a population level, we need information across these scales of biological organization. And I think that will be illustrated to a certain extent in the talk as well. So back to this slide and my Bengal tiger, which has nothing to do with the presentation, but it kind of goes with the color scheme. So that's why he's there. So what drives this extinction risk, and how can the extinction crisis, and how can we figure out, like, what techniques can we use to really try to understand what the contribution of pollution in particular is to threatening or endangering wildlife? It's really difficult, right, in the context of all of these other factors, which certainly also have a role. I mean, habitat loss is really important in terms of threatening wildlife health. And it's a little bit more obvious in a way, maybe, than something like pollution or pathogens and disease, where you have to actually capture the organism, maybe measure either like the presence of disease in their tissues or the presence of contaminants, for example, and where like the cause and effect is pretty difficult to sort out, especially in the context of all of these other factors that are happening. And not to mention that it's really difficult to do this work with endangered species. And so today I'm going to sort of show you some of the approaches that we've used to try and address this problem. One of the main ways that we study ecotoxicology in terms of determining what the effects are on wildlife is through field studies. So this is an example from like a different project that I've worked on for a number of years, which is in the Great Lakes, so shared between the US and Canada. And the species that we're using there is the herring gull. It's a colonial fish-eating bird. And all these numbers here are different colonies of these herring gulls across the Great Lakes that have been monitored for decades in terms of the health. So how many, some markers of health in the adults, but also how many chicks are hatching. And importantly, the presence of contaminants in the gull's actual eggs. So this illustrates the idea of using wildlife as sentinels. So, can we look in the environment and over time monitor how these populations are doing to try and alert us to problems with contaminants, either at certain areas or across like an entire ecosystem. Another approach that we often use in this context is to compare a control site to an impacted site. So for example, site 6 here in Lake Huron has often been used as a control site. It's far away from industry, and so the levels of contaminants in the eggs of those birds are a little bit lower. And then comparing what you see on a colony 6, for example, versus maybe some of these colonies in Lake Ontario or Lake Erie that are close to the cities of Toronto, Detroit, and have a lot higher contaminant levels. The problem with this, of course, is that really, what do we mean by control in this context? There is no control when you look in a natural population in terms of evolutionary times, historical levels of contaminants. Even in the most remote site in the Arctic, you will find many anthropogenic contaminants in birds' eggs. And so this comparison just, becomes messy and difficult in the context of not being able to find a meaningful control. In fact, I think this site, they've switched from that one to trying to use another one because those eggs were too contaminated. The other, so this is sort of just an idea of how we, tend to do like ecotoxicology in the field, comparing control and contaminated sites, doing biomonitoring over time. But the other approach, which my lab is also quite invested in, is to do lab-based studies and toxicity testing. And this is where you take a standardized model species. So for example, for fish, often fathead minnows are used in the top photo. And for birds, bobwhite quail, Japanese quail, chickens are often used for these tests. And so you take a group of these animals and then you expose them to a dilution series, so graded concentrations of the chemical that you're interested in. These tests, again, are standardized internationally, so you can compare between labs. And then you monitor effects like growth, reproduction, and survival. And you get information about what level of that single chemical is, quote unquote, safe in those species. There's some problems with these tests. One is ethical concerns, is that they use a large number of animals to accomplish. Another is that they don't give information about more subtle outcomes. Things like endocrine disruption are only just starting to be captured with these standardized tests, but there's a lot of effects that could affect species at a population level that aren't captured with this rather blunt instrument. Sometimes these are called kill 'em and count 'em tests because they're really quite extreme in that sense. A major challenge to all of this, which is very relevant to today's topic of endangered species, is that both of these approaches, so using the gulls as a sentinel species, for example, or using the fathead minnows or the quail as a model lab species, they really only capture information for that particular species. And what we know is that sensitivity between species varies extremely widely among species. So here's just one example of a species sensitivity distribution for a chemical called BAP, which is benzoypyrene. It's a polycyclic aromatic hydrocarbon. Doesn't really matter what the chemical is. The point is that these LC50 values, so these are lethal concentrations for 50% of the animals in that study, vary by many orders of magnitude between different aquatic species. And so what you find out for one species is not necessarily applicable to others. And you can see how this could start to be problematic, especially when we're working with endangered species for which we certainly don't want to be conducting toxicity tests. I have an example of that just to kind of drive home the point from some of my previous research. This is actually goes way back to some work I did for my PhD, although I'm still kind of dabbling in this topic. So the work we did showed that chickens, which is a model test species, are much, much more sensitive than other species like the herring gull, the biosentinel species I mentioned before, to a class of chemicals called dioxins. So the LD50, that's the lethal dose for 50% of the individuals tested, is 0.15 micrograms per kilogram for the chicken, so quite a small concentration, and over 50 micrograms per kilogram for the herring gull. And the reason it's more than 50 is that somebody tested up to 50 and saw no mortality. So it's even higher than that. Chickens are in fact more than 300 times more sensitive than herring gulls to dioxins. Now this is very consequential because at current levels of dioxins in herring gull eggs are range from 0.07 to 1, maybe across those different colonies that I showed on the map. And the LD50 for chickens is within this range. So what this suggests is that while herring gulls are currently at current levels are okay, they're not going to, their chicks are not going to experience embryo mortality due to levels of dioxins in their eggs. There could be other species out there that are as sensitive as chickens that would surely not be living in the Great Lakes anymore with concentrations of dioxins at these levels. And yet these are probably not species that we're going to give a lot of attention to. We might list pollution as a potential cause for the fact that they're endangered or threatened on a list, but we won't have that kind of smoking gun proof that actually dioxins is what killed those species. So just an example of how species sensitivity is really actually very consequential when we are looking at trying to be protective of wildlife in the environment. So all this to kind of say that it's important to think about and study endangered species, although we don't very often in the field of ecotox, because these are species that may actually be the really sensitive ones. They may be the ones that are the most impacted by contaminants, but they're going to be the least studied because a lot of our studies as ecotoxicologists involve sacrificing the animals, right? If you want to measure the levels of contaminants in tissues, you have to sacrifice the fish or the bird in order to get those tissues. If you're going to do toxicity tests, there's definitely a lot of euthanasia involved there. So information about endangered species is really lacking in terms of their sensitivity to toxicants. So I'm going to now switch into like the actual topic of the talk today and tell you about our approach for addressing this problem and how we're using molecular biology and early life stage tests to kind of get around some of the issues with working with endangered species. So just in general, like, okay, we're working with early life stage, that means like developmental models. So in general, like, is that okay? Is it, is a good life stage to work at? Or are we actually interested in toxicity at early life stages? And I think the answer to this is definitely yes, because often the developmental stage is most sensitive to contaminants. It also costs a lot less and it takes a lot less time to do these toxicity tests on early life stage organisms. And many would argue that it's more ethical than using adults. For fish, there's a great opportunity to work with endangered species because of the huge numbers of embryos that some species generate. So, some female fish will spawn tens to hundreds of thousands of eggs. And so even for an endangered species, you take 1,000 embryos from that, you're not going to have a significant impact on the population. And that's the approach we used here with the Copper Redhorse. So finally, introduce our study species, which is the Copper Redhorse. It is, here's a picture of it. It's the only vertebrate species that's endemic to Quebec, and it is indeed endangered. There's less than 2,000 individuals living in the wild currently. One of the reasons it might be endangered is it takes a long time for it to reach sexual maturity, so almost about 10 years. And currently, there's only 2 known spawning grounds for the Copper Redhorse, and they're both on the river I showed you, the Richelieu River. We did this study with Copper Redhorse in mind, but we also used another species. I don't have a picture of it because it looks kind of identical, except Hugo, my student, would be able to tell the difference, but they're very similar looking. So this is the river redhorse, closely related species, same genus. It is a species of special concern, so that's one level up from no concern. And so it's doing a lot better. It takes less time to reach sexual maturity, and there are populations in Quebec, Ontario, and also in the US. So we want to compare these two to see maybe if there's a difference in sensitivity between the two species. The site, again, as I told you, is here. It's the Richelieu River. And it is, it flows between Lake Champlain and the St. Lawrence, and it's a highly agricultural area. So it's surrounded by municipalities, but also it's a watershed for a lot of agriculture. And there are many tributaries into the Richelieu River where the concentrations of agricultural pesticides are above CCME threshold. So these are threshold levels that are set as being safe to aquatic life by the Canadian Council of Ministers of the Environment. So there is a concern with levels of pesticides in this river. Here's a picture of the river, and you can see that, and this is typical, that the fields go right up to the road, which is right next to the river. So you can imagine that after it rains, when pesticides are applied, like in May, it rains, and then those pesticides just get washed right directly into the river. Here, for example, is a picture of a farmer spraying pesticides in the field less than 1 kilometer from the river. It's not just pesticides, so there's other contaminants, and I'll talk about them a little bit later in the talk as well. So there's 21 wastewater treatment plants that feed into the Richelieu River, and there's numerous golf courses as well. So, you know, as most rivers, unfortunately, in our urban landscape, it's just full of a mixture of different contaminants. So the aims of this project were, first of all, to figure out whether the water itself is toxic to the fish, to the developing fish. So does Richelieu River water negatively affect the development of these two species, the copper and the river redhorse? The next aim was to try and figure out, okay, if we do see effect of the water on these fish, then what is it in the water? What component of it? What contaminants in the water might be contributing to these observed effects? And then finally, the last aim, which we're still working on, is what are the long-term consequences of exposure to this water? Is this early start to the copper redhorse embryos in this highly contaminated environment, is that really contributing to their inability to reproduce and create, sustain the population? And so that part we're obviously not using the species itself for. We had to use a model species for aim 3, which is zebrafish. So I'll get to that at the very end of the talk. Okay, so here is just the river again. It's flowing this way, this direction northwards to the St. Lawrence, and there's the two spawning sites. One is Chambly Basin, and the other one is at Saint-Ours. So these are the two known sites where the copper redhorse has been observed to spawn. There's no other sites that we know about in the world. The study itself, the first one that I'll talk about, took place at the Saint-Ours site. So this is the downstream site, also meaning that all of the contaminants from the watershed end up in the water at Saint-Ours, so that would be the more impacted site. So the Saint-Ours site is at a dam, so you can see it here in this picture. The fish spawn in the rapids below the dam. And there's also, this is not the best picture, but there's a fish ladder here, which again is shown here. And so the MFFP, the Ministère des Forêts, de la Faune et des Parcs, very long government acronym. MFFP is a provincial government agency. So they run an artificial breeding program right here at the Saint-Ours River. There's actually, they have a little field station right under the dam, like under the bridge here. So what they do is they capture the fish as they go up the ladder, the ones that are going farther upstream, maybe to Chambly to spawn. And they also fish from boats in the rapids themselves. And so they try to capture every copper redhorse, male and female, that's coming up the river. So when they capture the fish, they'll bring them into their little riverside lab, and they either wait till they're naturally ready to spawn, or they'll inject them with hormones to get the eggs and the sperm. They have a bunch of cryopreserved sperm, and they also have used microsatellites to genetically identify all of the individuals that they catch. So they have this really impressive pedigree of the individuals that they have caught for the past 10 years of this program. And then they use they artificially fertilize the eggs, and then they take them to another facility to raise until a certain juvenile stage when they're re-released into the environment. So all we did was we're able to get 1,000 fertilized embryos of each species from this MFFP organization in order to run our studies right in the research facility right beside the river. And since it's only 1,000 out of 100,000 eggs, this was just a negligible effect on the breeding program. So they were happy to participate in this with us. So we get the fertilized eggs, and then I'll show you a diagram in a minute, but we kept them in these tanks, control tanks and river water tanks in the riverside facility, so right under this bridge here, right next to the river. The eggs the embryos that we got were from the river and copper redhorse. River redhorse tend to breed a little bit earlier in June, copper redhorse a little bit later, but both are going to correspond with that peak of pesticides in the river. Pesticides are applied in May and early June, and then washed into the river when there are rain events. So in addition to the actual experiment, so this was in 2018, so we were able to do the river redhorse exposure first from the 9th of June to the 23rd of June, and then the copper redhorse exposure from the 15th to the 29th of June. And so this would take the embryos up through the larval stage just prior to swim up. In addition to doing these exposures, well, I'll show with the fish, which I'll explain in a minute, we also had passive samplers. So these are devices that we put right in the river that are kind of mimicking like a fish gill, and they will be collecting over time all of the contaminants that are flowing past them in the river. So they integrate the contaminants that are present over the 30 days that they were left in the river, these passive samplers. So we did some chemical analysis from them. And we also had daily grab samples. So that means we took water from the tanks where we had the fish and also directly from the river itself. So this is a little diagram of the setup, which was very ingeniously thought up by Hugo. So what we did was we piped water in from the river, and then it collected in a settling basin, which isn't shown on this diagram. And then it went directly into the tank, then out of the tank and back out to the river. And inside the tank were 3 tumblers. So these were just little plastic vessels with aeration, and the eggs were placed inside, the embryos were placed inside the tumblers, and the aeration made them tumble. That's why they're called tumblers. And that kept the embryos very viable, no fungi or any problems with viability. So that's the river water exposure. So those embryos are just being exposed to whatever day by day is flowing down the river for the 14 days of the experiment. And then the lab water control, we had a smaller tank placed inside the larger tank. Again, river water is being piped in, but inside the small tank is lab water. So clean water that's matched to the river water for things like pH and conductivity. And then the outer chamber had the river water, which maintained the same temperature, which was, this is in the springtime, is increasing daily throughout the experiment. So here is the first data I'll show you, which shows the lab water tanks, the river water tanks, and then the actual river itself, and how they match for these different water parameters. So temperature matched really very closely between the 3. That worked out really well. Dissolved oxygen was quite close, as was pH. Conductivity, we had a problem. So in the lab water tanks, there was an error made at the beginning of the experiment with the calculation. So that's why those values are completely off in the lab water tanks versus in the river water in the Richelieu River. And then turbidity was variable and spiked after the rain, and that's to be expected as all sediments are brought into the river. And then in the lab water, it was really non-existent. I will say that the difference in turbidity between the lab water tanks and the river water tanks was not as great as shown on this graph, because turbidity was measured in the outer tank, not in the tumblers themselves. And the tumblers had a little filter, so the eggs were not exposed to as much sediment as would have been in the outer tank. But in general, other than conductivity, we had a really close match between these water parameters for the treatment and the control. Survival was really amazingly high. This is the first time we did this, so we're very, very happy. We've had other experiences with embryonic and larval fish, which were not nearly so successful. So the river water did decrease survival in, this is the river redhorse species first, so the one of least concern. So the river water did, that treatment had lower survival, but it wasn't statistically significant. And also just survival was above 85% for all of them. So really high. The copper redhorse is an endangered species now. It had very similar survival throughout most of the experiment for the river water and for the lab water. But then right at the last day of the experiment, there was this big drop in survival in the river water treatment. And, we had to take down the experiment at that point for various logistic reasons, but it would have been really interesting to continue and to see what was happening there, why there was this sudden decrease. And we still don't really know, but we would have to do some more research, and May this spring, if all goes well, to try and figure out, what's happening with that data point. Hatching was quite interesting. So with the river redhorse, again, the species that has much stronger populations, the river water treatment caused slightly early hatching. But for the copper redhorse, this effect was really pronounced. And these data are just by date, but Hugo also expressed it by cumulative degree days, so keeping taking into account the temperature difference, because the river redhorse were initially at colder temperatures because they started a little bit earlier in the season. And there's a significant difference in this hatching rate for the river water versus the lab water. And this was really interesting because pesticides and some other contaminants as well have previously been shown to alter hatching, whether it's to hatching times, whether it's to make them earlier or later in different species of fish. And so this is perhaps some indication that maybe we should be looking a little closer at pesticides to explain this effect. We also looked at deformities, and we saw very small incidence of deformities. Here's a couple of clearly deformed larvae, and there were no significant effects of treatment, so nothing there. As I mentioned, we're primarily a molecular lab, although this study has more fieldwork and more organismal level endpoints. But one of the techniques that we used to try to understand what was going on was RNA sequencing, which is a method that captures gene expression across the whole genome. So it gives you an idea of like all the genes that are turned on and off and at what levels in a treatment versus a control setup. So we did this comparing just for Copper Redhorse because RNA sequencing is very expensive. So although the prices are coming down, but this was a couple of years ago. So very expensive. So we compared the lab water control to the river water. We had 9 replicates. That's each of those 3 tumblers in 3 tanks. And in each sample, we actually pooled 5 larvae as well. This work is super challenging to do with any non-model species because to do RNA sequencing, what you get back from the method is lots of little snippets of nucleotide sequences, which is great. But if you don't have a genome to compare them to, then it's really hard to know what they are and what they do, right? So Copper Redhorse had never been studied in this manner before. So there's no genome put together. There's no genome sequence that's known. And more importantly, perhaps there's no annotation, meaning when we have a little chunk of nucleotide sequence, we don't know what gene it belongs to. And so luckily, we have some very intelligent and generous bioinformatics friends, and they worked on a project with us to develop a tool called Seek2Fun, which is a publicly available tool which really addresses this very problem of working with non-model species. And what it does, instead of going to the nucleotide sequence, which is what you would do with a model species, like if you're working with a mouse or a zebrafish or something where the whole genome's known and we know which genes correspond to which sequences, rather than doing that, you translate the little snippets of DNA into proteins and then use a protein database that is more conserved across all different species to understand which genes are turned on and off in your samples. So, to explain all that, well, I probably couldn't do it properly and it would take several hours. But suffice it to say that this is a really innovative tool that uses protein sequences to do analysis in these species for which we don't have the right information about their genomes. So we were able to successfully do this, which was great. We generated the first transcriptome for this Copper Redhorse species. And we saw 11 differentially expressed genes between the 2 treatments, between the river water and the control. This is a low number. So if for example, we had exposed the fish to like a high concentration of pesticides, you'd expect to see like hundreds of genes changing. So this suggests that while there are some effects happening, that they're not extensive across the genome. But we did see upregulation of cytochrome P450 pathways, which are involved in xenobiotic metabolism. So that is a sign that they are being exposed to contaminants and they're at levels that are causing a biological response. And also downregulation of immune function pathways. And so that would be just something interesting to follow up on in terms of a hint of what types of effects to look at the organismal level in follow-up studies. So that's it for the RNA sequencing results. We also, as I mentioned, used passive samplers to try to detect what pesticides were in the river. And we detected all these different pesticides with the passive samplers. This list doesn't include any neonicotinoids, which are pesticides which you've likely heard of and are currently being used quite a bit in the area. Just because of a technical analytical chemistry limitation that they can't get those data from the passive samplers. Just as an example, though, from like this was sort of our first effort to detect, what chemicals were in the river that could be causing this early hatching. So for atrazine, as one example, we saw an average concentration from the passive samplers of 164 nanograms per liter. So that's allowing for the fact that the passive sampler was sitting in the river for 30 days. So it's kind of like an average of what was flowing down that river over that month. And then we also have these grab samples on different days in June, which is just a snapshot of the concentration of atrazine on that one particular day. So on the 16th of June is 25 nanograms per liter. The 19th of June, it was 560 nanograms per liter, and then back down to 12 on the 29th. And this all makes perfect sense because there was a huge rainstorm shortly before the 19th of June. And so that's why you get this spike of atrazine in the water at that time. And also sort of makes sense that the average, would be around 164. These levels are not super high, but 500 nanograms per liter. So the concentration after the rain has been shown to have a negative impact on reproduction in that model species, fathead minnow. And 25,000 nanograms per liter, much higher, has been shown to cause early hatching and emergence of sockeye salmon larvae. I also want to point out here the CCME toxicity threshold, which is 1,800 nanograms per liter. So just to highlight that this is not necessarily a level that is safe for larval fish. It's just a general level that's considered to be safe for aquatic life. But if larval fish haven't been tested, then it's not going to be included as part of that safety threshold. So that CCMA guideline needs to be taken with a grain of salt. It's decided on the best data available, but they may not be very applicable to the system you're looking at. So following up on that, we did a lab pesticide exposure with, this time we added walleye, but also river redhorse and copper redhorse, where we, this was the next spring, we got the embryos again, but this time we want to control what they were exposed to. So let's see if we expose them to some of the pesticides that we think are causing this effect, do we get the same results that we saw in the field? And this is to try and kind of make sense of that multi-stressors, complex mixture of contaminants scenario. So we chose atrazine, glyphosate, and metallochlor, which were 3 of the chemicals that we detected with the passive samplers and were known to be high in the river and also based on our data. And clothianidin, which is a neonicotinoid. And we tested 3 concentrations. One, the low is the Richelieu River, just sort of standard level. The medium was the level that's found in tributaries to the river, where the concentrations are generally higher, and then 10 times the tributary. With these tox testing, you want to have an environmentally relevant concentration, but also a higher concentration so that you can see what kind of effects to expect at higher concentrations. We did not see much. We didn't see any effects on hatching, deformities, or mortality in walleye and river redhorse. And unfortunately, in that year, we got very poor copper redhorse embryos. They only got 1 fish, and we had only 10% survival. You can see here the river redhorse and the copper redhorse they should be the same size, but the embryos we got for the copper redhorse and the larvae that did hatch were much, much smaller. And so those data were really not useful. We'd planned to repeat this this year, but COVID, so we didn't get to do it. And hopefully we'll be able to do it next year and see if the copper redhorse are maybe more sensitive, as we saw from in the first year's exposure to these pesticides, which would be quite interesting. So we have some organismal level effects, but we are not sure what's causing them. So far, the treatment we have is just the river water. So what is it in the river water that's causing that? So we did some more chemistry to try and sort this out. And here we use the Chambly site as well as Saint-Ours. If we blow up the site, you see that there's the Chambly Basin. This is the spawning site of the fish here. We collected water there. And then downstream is the Huron River and the Acadie River. So two different tributaries that have higher concentrations of pesticides because the ag fields just feed directly into them. So we did Chambly, Huron, Acadie, and Saint-Ours. In this case, we collected samples almost daily. This is during the same year when the copper and river redhorse were exposed riverside. And then we did some really extensive chemical analysis to look at the trends through time in these concentrations of chemicals. Part of this analysis was a targeted pesticide analysis. So we targeted 9 different herbicides, neonicotinoid insecticides, and AMPA is a degradation product of glyphosate, a very popular pesticide. And these are 9 compounds that were previously detected in the Richelieu River and tributaries. They are the ones that are most often detected and at the highest concentrations. I won't show you all of the data because that would take too long, but just some kind of highlights. There you go. So atrazine, you can see at Acadie and Huron, so those are the 2 warmer colors. Those are the tributaries. So they're going to always be at higher concentrations. So they were quite high. And Saint-Ours is higher than Chambly as we expected. Chambly is the upstream site, Saint-Ours is the downstream site. You can also see really clearly here how after the rain event on the 18th, shown in blue, that's when the pesticides spike in the river. So that's atrazine. Glyphosate similarly spiked, same pattern, and spiked after the rain event. Both of these are way below what we would expect to see toxic effects. But some of the other chemicals like thiamethoxam, which is a neonicotinoid, we did have levels above the toxicity threshold. Here it's 8.3 nanograms per liter, shown by this black line at Chambly and Saint-Ours. So that's within the river itself. And if we change the axis, you'll see now that these are really much, much higher in the tributaries. So those are being pumped into the river at very high levels. Same thing for clothianidin, which is one of the pesticides we tested in the lab in 2019. So again, Saint-Ours, the levels are above the toxicity threshold and much higher in the 2 tributaries. In addition to these targeted pesticides, we were able to detect 4,291 other compounds. So when I say we, this is the analytical chemists that we're collaborating with. For the targeted pesticides, you have to run an analytical standard along with the pesticide. You have to know what you're looking for, be able to quantify it. For these untargeted ones, they can just tell us if it's there or not, and if it's different between like the Saint-Ours and the Chambly site, for example. So out of that, we have identified 342 of these 4,291, and 41 have been validated. And the unidentified compounds are probably either just absent from the database, like we just don't know about them, or they're natural compounds and probably not of too much interest. So this just, I think, really illustrates, and I'll kind of quickly go through some of the compounds that we found in the river that, we're focusing on pesticides, but there's a lot of other stuff in the river as well that could be causing some of these effects. So other pesticides, for example, than the ones we targeted were detected at higher concentrations at Saint-Ours than Chambly. Pharmaceutical and personal care products, we had 22 compounds, all kinds of drugs like acetaminophen, antidepressants, estrogen, cancer treatment drugs. Also, some food additives and flame retardants were detected in the water. So it's complicated. Trying to find out which chemicals and what components of this mixture are causing any effects that might be happening is really complex. I'll just finish on the final aim, which we're in the process of doing, and that was to try to understand what might be happening in fish later in their lives. So if we expose the embryos and the larvae to this river water, is it really going to have an impact on the population? Is it going to prevent these adults from reaching reproductive maturity at 10 years old, or will it reduce their reproductive maturity? This, of course, would be pretty impossible to do with the actual copper redhorse. So we turned to our zebrafish model for this. And what Hugo has done is expose the eggs and the embryos and larvae to river water that we actually collected from Chambly and Saint-Ours, so 2 different concentrations, and also the control lab water. And so far, he's hatched the larvae, he's growing them up in the lab. He did see altered hatching times with the river water, so that was quite interesting. And now we have to wait a few months until these fish are reproductively mature. In zebrafish, it only takes 3 months rather than 10 years. And we're going to assess their fecundity, so their growth as well, to see if that experience early in life was altering their reproductive output. I should mention that after the larval stage, we're exposing them to clean water for the rest of the life cycle. So just to conclude and kind of summarize the results from this study, the most striking result from an organismal level was the early hatching, which I think is quite interesting, and its significance to the population. It's kind of hard to say, but it could have an effect depending on well, it could have an effect on the population. We also need to kind of finish up our study to resolve this question about species sensitivity. It'd be really interesting to see is the copper redhorse actually more sensitive than the river redhorse? That would be a very interesting finding to some of these pesticides. From a molecular level, we found that metabolism and immune response may be affected. And the chemistry told us that there are really a lot of different things in the river. And that the pesticides, we knew this, but to see it makes it quite interesting that they really spike after the rain events. And, that's going to happen at different developmental stages and different years. And how does that just quick, but really acute exposure to a pesticide during development, how does that affect the fish differently at different developmental stages? So there's definitely a lot more work to do to sort of answer the question whether contaminants are contributing to the species decline. But we think we have a good approach and some good tools to work with. And with that, I'd like to just thank the funding agency, which is Fisheries and Oceans Canada and the National Contaminants Advisory Group. And really to thank everyone on this list for contributing to this project, but particularly the staff at the scientists at the MFFP, who we couldn't do any of this without them. They're the ones who are running the breeding program and provided us with the embryos. And then of course Hugo, who's done a great job with this project and is writing it all up as we speak. And with that, I went a little longer than I expected, so I would be really pleased to answer any questions that have come up.
Great, thank you so much, Dr. Head. This was really, really informative, really interesting conversation, really interesting study. So I think that at this point, we do have several questions, or a couple questions that have come in, or at least, and so Marie, would you like to read the questions?
Sure, I can read the questions. Oh, maybe I can. Okay.
Can you see the chat there?
Yep, I got it. It did something weird when I opened it and was like a blurry mess, but now it's clear.
And then of course, for anybody who is listening in the chat or feel free to enter your continue to enter your questions into the chat. Dr. Head will stick around for a while to ask a few questions. So, so if you've got questions, good time to ask them.
So I'll start with one from Brylan. Are there any current programs aimed to educate the public, especially the farmers, to try to reduce the runoff of the pollution in that area?
Educated farmers, that I'm not so sure about. There's definitely there's definitely a lot of talk about banning certain pesticides. So that I think is where the action is. Farmers generally, I mean, they do tend to care about the environment, you know, but they also want a product that works and that they can, that's cheap and effective and safe. And to my mind, you know, the issue with this is not so much, is not, you know, if you want to make a big impact, it's not so much educating the farmers as much as reforming our system of regulating these pesticides. Because what often happens is, you know, a study like this one will say, oh, this certain pesticide is really toxic to this endangered fish species. And then there'll be this political and public will to ban that pesticide. But what happens after that is that a new pesticide comes on the market. And the thing about the new pesticide is that we don't really know anything about it. And so then people like me do research, and then 10 years later we say, oh, this one's bad, so let's ban this one. And so it just goes round and round and round and round. And that's, I think, what's really frustrating about this field. Like, for example, glyphosate is a pesticide that the public is really very much against. At least, well, I don't know, that's based on my Facebook page. Maybe it's not generally true, but it's really not that toxic compared to some of the even new ones that are on the market. So there's something really broken about the way we regulate these things. That's not quite answering your question, but maybe sort of adjacent.
Yeah, great. There's just a comment about sad to see all the human pharmaceuticals in the water, which I guess is not that surprising, but yeah, that was a lot, right?
Yeah, and the thing about those is that they are designed to be bioactive. So, you know, even a small concentration, like those things are designed to interact with vertebrate receptors and enzymes. Yeah, there's a diabetes drug, I don't know if it's on that list, but that's been found like in some US rivers at incredibly high concentrations. So that is a little disturbing, yeah.
And there's some more questions. I'll keep going. Because this is from Shelley. Because genes are an important aspect of the project, how did you choose the embryos? You would likely need representative from several females and males.
Yeah, so this whole experiment was from one, they're all siblings. So that's really important. I shouldn't mention that because that's really important. So, you know, the differences we see between species could just be a genetic difference because of the parents. So for the study, in a sense, you know, we didn't really have a choice about that because it's an endangered species. Like, we were lucky they caught a female and they were able to give us embryos. But yeah, it's all from one cross. So all the Copper Redhorse are from one cross and all the River Redhorse. Which is nice because then the siblings are all like fairly genetically uniform, so we can kind of pick out the effects of the contaminants without having to worry about like the quality of the parents. But yeah, if we're looking at species difference, it's a bit of a bold claim to say that the, you know, Copper Redhorse is more sensitive than the River Redhorse. Maybe it's just like Susie Copper Redhorse is more sensitive than Sarah River Redhorse. So yeah, that's a really important point.
Yeah, great. And Shelley just followed up to say that, yeah, she was wondering if there were these different pros and cons of the genetic diversity of the embryos that you selected, so that you answered her question, I think.
Okay, good.
So we've got another question from Jim. Are you aware of negative effects of mosquito control chemicals used in residential lawns, near aquatic areas, upon a North American fishery species.
Like any particular ones? What's the mosquito control chemical? I'm not so sure. Do you know, Marie?
I don't know that I would know off the top of my head which kind of pesticide that would relate to.
I've never heard any. I mean, that doesn't mean. Yeah, I'm not sure. That's a good question. Like,
Yeah,
bees or something like that. You mean like just to keep off?
I don't know. Is that maybe it's not something that they do very much of in Canada? So, well, I mean, I know in Louisiana they would just like spread that stuff literally everywhere, like whatever. I don't know what chemical it was off the top of my head, but there would be like this mosquito control trucks that would like drive down the roads there and just like spray
Like
something.
DDT, I guess.
Yeah,
DDT
yeah.
replacement. Yeah, I don't know. That's a good
Yeah.
question. I'm not aware of what chemical that is, but it
Oh,
probably
yeah.
has an effect on aquatic species.
Yeah, I don't know either. Shelley had some suggestions of permethrin, or I'm not going to pronounce that. Shelley, do you know how to pronounce it?
Yeah, I was just thinking permethrin or deltamethrin. I was just getting ready to send a chat saying that Sean Strom, if he's still on, he might know. He's our a toxicologist in the state. I don't know if Sean had to leave, but
I think I saw it on my list. Yeah, I'm not so sure about that one.
Nope. Yeah. Good question though. I was curious about what you guys used as passive samplers for the stream. Were those just well, I was just curious about them in general.
It's yeah, my memory might fail me here. It's POCUS, is that what they're called? So I forget what the acronym stands for. It's just like a little canister with there's different ones depending what you're trying to capture. It just has a membrane in it, which captures all the chemicals, and then you take out the membrane, you extract everything that's on it. And then the tricky part is you have to have these constants that describe the relationship between what's in the river and what would be captured on the membrane. So actually, validating the exact concentrations is difficult, but it just kind of, it was nice in a way because it gave a snapshot of what came down the river over that 30-day period, right? Because like it's a lot cheaper. It just integrates everything over the 30 days. So you don't have to measure every single day. Yeah. Because as you saw, there's like a lot of variation from day to day.
Yeah, I like the idea, and I was just curious. Did you get those commercially? Were they commercial?
Yeah. And then we sent them to Access Analytical to analyze them. So not everyone can do the analysis either. It's
right.
Kind of specialized. Yeah.
Yeah.
Yeah, there's all different ones for like depending what types of compounds you're trying to capture.
Cool. So there's a couple more comments. The question related to the mosquito control was just that he'd been told that chemicals used are proprietary mixtures by the company, which would not surprise me.
So maybe some of those unidentified compounds.
Yeah. And then Sean did pipe up that some of the mosquito control is via oh gosh, I cannot pronounce this, but BT, the Bacillus I'm not going to pronounce it right. I know what it is. Yeah, I'm not going to do that well, but it's not a pesticide. It's a biological control.
Oh, okay.
Bacillus thuringiensis.
Valiant effort.
That's about how I would say it as well. Yeah. And there's a question about what are the main crops in the area, and also is the early hatching linked to poor health in fish?
The crops there's corn, there's soy. I think that's the main crops. I know they're using a lot of glyphosate still, and there's some neonics. It's not early hatching. Let's see, I'm trying to think what Hugo told me about this. I think there's what he was saying is there's an issue, like a ecological issue, and maybe some others on this call. You guys are wildlife department, you might probably know better than me, but there's an issue if the hatching takes longer. Like if they all hatch at once, then they can escape predation, whereas if they hatch in little bits and bobs, then they're more likely to get eaten. So that's one factor. I'm not remembering what we discussed as the consequences of early hatching. Does anyone know who's on the call? What could be the consequences?
I don't know if we have any fisheries people left on the call or not.
Yeah, I should. But that's an important thing to consider for sure, because that's like the main effect we saw. So is it, just because you've seen effect, it may not have any bearing on the population.
Yeah, Shelley suggests that if they hatched earlier, they would be bigger before winter, maybe.
Because they start eating sooner.
Yeah, have more time to grow.
Yeah.
That is all the questions I had. If anyone else on the call has anything else, feel free to type it in the chat. But I think that's all that we have currently.